one of the urologists at City of Hope. I'm excited to co-moderate this session with Dr. Varner. The last talks are really exciting. We have a mix of clinical, preclinical, translational, and new drugs, so I think it's really worth staying into the video. The first talk's going to be from Dr. Giuliana Mognol from UCSD, defining systemic and tumor-intrinsic biomarkers of immune suppression in RCC.
Good afternoon. Thank you for the opportunity of presenting our work here. RCC has a highly immunosuppressive tumor microenvironment enriched in tumor-associated macrophages and Tregs. Accordingly, high macrophages and Treg infiltration is associated with poor patient prognosis. Biomarkers that capture this immune suppression are still lacking. Our goals are to identify biomarkers of immune response and suppression in paired tumors and blood, and evaluate immune suppression target therapies in patients-derived tumor slices. So to identify biomarkers in RCC, we apply this integrated multi-omic analysis where we use blood from healthy donors and paired blood in tumors from cancer patients. From the blood, we profiled the immune responses, the systemic immune responses using serum proteomics, FACS, flow cytometry, or single-cell RNA sequencing, and we analyzed the tumor by IHC and single-cell sequencing. This study is ongoing, and our aim is to integrate all data from blood and tumor to identify strong biomarkers to predict immune response and suppression in RCC. From our tumor cohort, we identify high macrophage and T-cell infiltration compared to the adjacent normal kidney, with a trend towards an increase in macrophage and decrease in T-cell infiltration in late tumor stages. Can tumor-associated immune suppression be detected in the circulation? So in peripheral blood, we show an increase, an expansion in Tregs, and a decrease in activated monocytes that are HLA-DR positive, indicating immune suppression and decrease in antigen presentation. Do circulating proteins reflect these RCC alterations? So using high-throughput serum proteomic analysis, we identify several proteins that are upregulated in RCC, including IL-6, KIM-1, and FABP1, consistent with systemic inflammation, showing renal injury and metabolic alterations. Our GSE analysis indicated several pathways that are alterated related with metabolic regulation and inflammation. So can we identify the cellular source of these systemic signatures? We are in the process now of analyzing our single-cell sequencing from PBMCs, adjacent normal tumor and tumor from these samples, and this will determine the activation and the exhaustion states of the immune cells, and also characterize the cellular interaction in the tumor microenvironment. This UMAP from our tumor cohorts confirms that the macrophages that are cluster number 1 in orange are a predominant immune population in RCCs compared to benign tumors. So can macrophage target therapies reprogram the RCC tumor microenvironment? We established patient-derived tumor slice cultures where freshly resect RCC tumor slices are cultivated in culture for 5 days in the presence of different therapeutics. We are evaluating macrophage target approaches such as PI3 kinase gamma inhibition to decrease the macrophages or repolarize the macrophages towards the stimulatory phenotype, and I'm happy to discuss this better further. As conclusions, RCC exhibits systemic immune dysregulation characterized by increased Tregs, reduced activated monocytes, and altered serum proteins. An integrated multi-omic approach will link pair systemic and tumor biomarkers to uncover immunosuppression and therapeutic biomarkers and targets in RCC. I would like to thank everybody involved in this project, my mentors, collaborators, and the patients. Thank you.
Thank you so much. So next we have Dr. Hans Hammers from UT Southwestern presenting on a novel oral PTPN inhibitor for cancer immunotherapy.
Great, thank you so much. I would like to thank the selection committee on behalf of my investigators to present some early data on, I think, a very interesting new small immune modulating drug which targets PTPN2. Next slide, please. Oh, I'll do this here. Sorry. So this is actually a very interesting target. This is a protein tyrosine phosphatase, which is intracellular, obviously, but in two compartments. One is the cancer cell compartment, the other one in the T cell. And it is really a master immune checkpoint in the sense that it really downregulates and dampens interferon signaling or other cytokine signaling in cancer cells, but also dampens the TCR response. And so targeting this, inactivating this phosphatase would actually reactivate these pathways. So really reigniting essentially interferon signaling and certainly reactivating or reinvigorating T cells. And this has been well published and was developed by Calico in combination with AbbVie. And what we will demonstrate today is some early data in the early data sets. This has been in clinical development for a little while. It's a challenging compound, or at least initial to work with, to get the right dosing and the right schedule. So these little boxes that you see here in orange are the boxes we're going to present some data on today, but believe me, this has been more complicated than it's indicated. After the initial dose escalation and schedule development, it went into some expansion with some heterogeneous tumor population, including non-small cell lung cancer, head, neck, and clear cell renal cell carcinoma. But in interest of this meeting, we will present on some of the data that we have in renal cell carcinoma. So these are the swimmer plots. As you can see, as you would expect, with immunotherapy, we do see a subset of these patients nicely responding with shrinking tumor, and the benefit can be quite prolonged. And in red, you see some data, which is really a small cohort in the current data set of monotherapy, and in blue is combination with PD-1 inhibition. On the next slide, you see two essential vignettes. I can't really read this well from here, but essentially these are patients who have been on immunotherapy for quite some time. One of the patients actually discontinued immunotherapy after 2 years and continues to do really well and is, quite frankly, potentially cured. So something that would go in the direction of effective, durable immune benefit. So coming back to this trial, we will essentially now commence, or are currently commencing or continuing expansions with the monotherapy in a certain schedule and dosing, and then also in combination with PD-1 inhibition. The top 5 of these side effects really characterize the side effect profile of this agent. So there are a few things to mention. Really, virtually all patients get some kind of fever, pyrexia, typically in the afternoon after they took their morning dose. They can actually be quite impressive, but can be managed well with Tylenol and NSAIDs. And with that, they get flu-like symptoms. And the other one that was kind of like one of the issues was liver function test elevations, which can be mild, but can also be quite impressive into the several hundreds. And it took time to realize that these are typically self-reversible. There's no need for steroids, and it's something that certainly affected the development. There were also no significant serious adverse events different between the groups of monotherapy, for example, and the combination. These are some preclinical data sets that encourage us to use or suggesting the potential combination with tyrosine kinase inhibitors. In this particular animal model, the combination with cabozantinib and 484 was significantly more effective than the single agents, and that is leading to the suggestion of another combination approach with a TKI, and that is still under discussion. So in conclusion, I do think this is a very interesting new target in immunotherapy, really first in class of an oral small modulator of the immune response, and we do see prolonged benefit, and the studies are continuing. Thank you so much for your attention.
All right, we'll move on next to Dr. Yufei Wang from Dana-Farber with a preclinical study of an anti-CA9 and CD70 CAR-T.
First, I would like to thank organizations for this opportunity to present our work on preclinical study of anti-CA9 CD70 deferred CAR T cell therapy, secreting anti-PD-1 CTLA-4 bispecific payload for advanced CCRCC treatment. So CAR T cell therapy has gained great success in the hematological malignancies. However, CAR T cell therapy has not been translated to the solid tumor treatment yet. There are several barriers that prevent CAR T cell therapy to translate to the CCRCC treatment, including antigen heterogeneity, on target of tumor toxicity, lack of persistence, of course, CAR T exhaustion, and tumor suppressive tumor microenvironment. So for the past decade, we have engineered different CAR T cell therapy, try to tackle these challenges, including collaborating with Dr. Sabina Scanradi. We characterized the CA9 CD70 distribution and expression on different stages of the CCRCC. We demonstrate the CA9 CD70 heterogeneously expressed in the CCRCC. So we engineered a bispecific tandem anti-CA9 CD70 CAR that can target either CA9 or CD70 positive tumor cells. And we performed the fine-tuning of the affinity and avidity of the CAR moiety to let the CAR only kill the tumor antigen high tumor cells, but not the same antigen at low levels on the normal tissues to mitigate the on-target of tumor toxicity. We optimized the different CAR design, and we found that 4-1BB co-stimulation domain actually outperform the other CAR constructs in our CCRCC orthotopic mouse model and achieved enhanced in vivo persistence. Last but not least, we armored our CAR T cells with immune checkpoint inhibitor. In this case, is a bispecific tetravalent antibody targeting both PD-1 and CTLA-4. And we have demonstrated this tetravalent antibody outperformed the EVO and NIPI, NIVO and EP combination therapy in an in vitro experiment. So we call this CAR T cells dual target fine-tuned immune restoring CAR T, deferred CAR, and we demonstrate that this CAR exhibited superior tumor killing in a multi-challenge assay. We compared this CAR with the same CAR construct with an irrelevant payload targeting cells. And we can see that with 3 different rounds, the deferred CAR T cells killed better compared to the B7-3 cells, and by maintaining the function and less exhaustion. Supported by the DoD, we are fortunate to start the GMP manufacturing of the lentiviral vector and GMP manufacture of the CAR T cells. Here I show the optimization of our CAR T titer and the protocol to elevate the transduction efficiency. Here I want to show that we were able to significantly increase the T cell transduction efficiency on the upper level. By optimizing the protocol, we were able to increase the transduction efficiency from single digit to like 30%. And after we increase the lentiviral vector titer significantly, we're able to achieve over 90% transduction efficiency currently. And this is my just last one, I want to show that we established a humanized IC15-DKO mouse model to characterize both efficacy and the safety of the different CAR T cells, and hopefully next time I'll be able to share more results. I would like to thank my mentors, Dr. Marasco, Dr. Freeman, a village of people who contributed to this work. Thank you for your attention.
All right, our penultimate speaker, Dr. Raul Winiak from City of Hope, prediction of immune-related adverse events in patients receiving first-line immune checkpoint inhibitor therapy.
Thank you to the Scientific Committee at KCRS for allowing me the opportunity to present my abstract on prediction of immune-related adverse events with hematologic markers in patients receiving first-line immune checkpoint inhibitor-based therapy in metastatic renal cell carcinoma. These are my key takeaway points. Frequency of IRAEs in patients receiving first-line ICI-based therapies for MRCC is high. Baseline hematologic ratio is strongly associated with first-line IRA occurrence. And further prospective validation of baseline hematologic ratios is merited. So immune checkpoint inhibitor-based therapies represent the foundation of first-line metastatic renal cell carcinoma treatment. Incidence of IRAEs in MRCC is high, at 44% for any grade, with first-line nivolumab ipilimumab conferring the highest rate of IRAEs. Grading by CTCAE of IRAEs guides treatment, with grade 2 and above warranting active treatment. And although prognostication based on IRA occurrence has been shown, prediction of IRA occurrence in MRCC remains a challenge. We use the City of Hope National Database to identify MRCC patients previously treated with first-line ICI-based therapies from September 2015 to September 2025. Patients were grouped into two cohorts, dual ICI or ICI-TKI. Using baseline levels of neutrophils, eosinophils, lymphocytes, and platelets, we calculated baseline neutrophil to eosinophil ratio, neutrophil to lymphocyte ratio, platelet to lymphocyte ratio, and systemic immune inflammation index, which is calculated by multiplying platelets and lymphocytes and dividing by platelets and neutrophils and divided by lymphocytes. We stratified patients into high and low groups for each hematologic ratio using recursive partitioning, and we evaluated association between baseline hematologic ratios and development of grade 2 and above IRAEs. Of the total 193 evaluable patients, 100 patients received first-line dual ICI regimens, and 93 received first-line ICI-TKI regimens. The median age was 64.2, and as you can see, histologic subtype and IMDC risk were fairly comparable across both cohorts. In total, grade 2 and above IRAEs were observed in 57 patients, with a median time to onset of 91 days. The incidence of IRAs were higher in the dual ICI group at 38%, contrasting with 20% in the ICI-TKI cohort. And the most common IRAs were ALT-AST elevations, observed in 18 patients. In multivariate analysis, we showed that all baseline hematologic ratios were significantly associated with development of grade 2 and above IRAs. Stratification of patients by ICI backbone revealed a selective association of baseline NER and SII with IRA prediction in the dual ICI cohort and baseline PLR in the ICI-TKI cohort. This may be explained by differential manipulation of the tumor microenvironment with each class of treatment. So in nivolumab ipilimumab, the tumor microenvironment undergoes systemic immune cell reprogramming. Therefore, low baseline NER and high baseline SII, reflecting eosinophil immune modulation and heightened systemic inflammatory state respectively, may correspond with IRA occurrence in dual ICI therapy, where toxicity is driven by immune dysregulation. On the other hand, TKIs alter the tumor microenvironment through modulation of VEGF-mediated inflammatory pathways. Therefore, high baseline PLR, which may reflect an inflammatory angiogenic phenotype and therefore high VEGF dependence, may associate more closely with IRA development in the ICI-TKI cohort, where the cells are basically undergoing endothelial damage, and that may predispose to the toxicity. So we've shown that frequency of grade 2 and above IRA is high in MRCC, particularly for dual ICI-treated patients. Baseline inflammatory ratios may capture different aspects of host immunity, depending on immune signature and ICI-based backbone. Early risk stratification of IRAs with personalized therapeutic monitoring and intervention for those at high risk. And further prospective validation of these hematologic biomarkers is required. So to summarize my key takeaway points, frequency of IRAs in patients receiving first-line ICI-based therapies is high, baseline hematologic ratio is strongly associated with first-line IRA occurrence, and further prospective validation of baseline hematologic ratios is merited. Thank you.
All right, our last presentation, but definitely not the least, a very outstanding meeting, Dr. Yue Chen from UC San Diego.
Good afternoon. I'm honored to be here on behalf of my co-authors, and I would like to thank the scientific committee for choosing this abstract for presentation. Oh, sorry, did I push it the wrong way? Oh, sorry. Okay, so it is well known to our audience here regarding the background science in kidney cancer, and ARN, also known as the HIP-1 beta, it is also essential HIP binding factors that it requires for transcriptional activities. However, ARN expression and its clinical relevance in RCC still remain to be defined. And as described by Dr. Tenza in her earlier presentation, that currently there is an ARN inhibitor being investigated in a phase 1/2 study, the NEO-811, and therefore it is important to understand ARN expression across RCC subtypes. So the aim of this study was to characterize an ARN expression across RCC subtypes as well as the molecular subgroups and evaluate its association with clinical outcomes. This is a collaboration with the Caris Life Sciences. So Caris performed DNA and RNA sequencing on RCC specimens. ARN expression was measured as transcript per million and dichotomized as high versus low quartiles based on quartiles. Overall survival was obtained from insurance claims data and calculated from initial diagnosis to death or less contact. Lastly, the VEGF TKI time on treatment is defined from first to last recorded treatment date. There were about 4,000 RCC tumors, including in our analysis. About 30% were clear cell RCC, 5% were papillary RCC, about less than 2% were chromophobe, and we had a few cases of medullary RCC. The rest were categorized as RCC NOS. 71% were male, about 61% were white patients, and we have about 68% of patients that were non-Hispanic patients. The median age of diagnosis in this cohort was around 64 years. Here we examine the ARN expression by histology subgroup as well as the tumor size. What we found was that ARN expression was quite conserved between all the RCC histological subgroups, with higher expression observed around clear RCC, papillary RCC, but lower in medullary and chromophobe RCC. The ARN expression is also quite consistent between primary RCC tumors, lymph node metastasis, and as well as the distant metastasis. On the right-hand side is the further breakdown of the ARN expression across tumor sites. Here we examine ARN expression by VHL alterations, as well as tumors with without sarcomatoid features. Again, we also notice quite consistent ARN expression in tumors with without VHL alterations, as well as tumor with without sarcomatoid features. Here we examine ARN expression across the racial and ethnicity subgroups, and again, we don't observe any statistical significant differences across racial or ethnicity subgroups here. Here we examine the co-occurring gene alterations comparing high versus low ARN expression. What we found was that tumors with high ARN expression has lower TP53 alterations, but modest increase in terms of the CHEK2 and PALB2 alterations. However, in terms of common RCC genomic alterations, including PBRM1, SET2, and also BAP1, we didn't notice any differences there. Finally, we examined the overall survival and VEGF TKI time on treatment by ARN expression. We didn't notice any association between ARN can be a predictive biomarkers there. So in conclusions, ARN expression is quite preserved across RCC histology, tumor sites, VHL alterations, or in tumors with without sarcomatoid features. We also didn't observe any differences in terms of expression across racial or ethnicity subgroups. High ARN expression is associated with more PALB2 and CHEK2 alterations, but fewer TP53 alterations. And again, ARN expression was not associated with overall survival or VEGF TKI time on treatment in kidney cancer patients. Our study suggests that there might be a crosstalk between the DNA damage repair pathways with the HIF-ARN signaling in kidney cancer, and prospective studies still required to validate ARN expression as a predictive biomarker in patients treated with ARN inhibitors. Thank you very much for your time.
Thank you for a great session. This is open for questions now. If anybody would like to ask a question, over here. Question.
Thank you so much for a great talk. I'd like to ask for first speaker, Giuliana, about the contribution of the peripheral to tumor microenvironment. I think that because the tumor express IL-6 or other immune or inflammatory molecules, so I just wonder the increase of Treg or decrease of activated monocyte result of these secretory molecule, or these increased Treg or decreased activated monocyte directly have something to do to the tumor microenvironment. I'm just curious whether it's more likely. And second question is, do you have any idea to prove which is more likely to be true?
To happen. Okay, so I think that for now we don't have the answer of that. We are still analyzing the results, but we have our cohort. Half of the cohort is early tumors, and we will correlate the data. So to see, for example, the patients that have more Tregs in the circulation or have more IL-6, are they early stage or they are late stage? So we don't have this answer yet, but it's known that macrophages, when they are polarized to the immune suppression phenotype, they can secrete cytokines that inhibit the immune response, and one of them is also IL-6. So we also can correlate the results with the macrophage percentage and with the macrophage phenotype in the tumors when we finalize the single cell data.
Thank you.
So this is a question for Giuliana. What is the rationale for this IPI 549 drug that you're using?
Mm-hmm.
Can you explain more about that?
Yeah, sure. So PI3 kinase gamma is expressed more in myeloid cells, so in macrophages. There is this small molecule that is in clinical stage, the IPI 449. The clinical name is eganelisib, if I'm not wrong with the pronunciation. So this small molecule binds to the catalytic domain of the PI3 kinase gamma and inhibits the ATP binding, so inhibits its function. So it will inhibit the function of the PI3 kinase gamma without decreasing its expression. So PI3 kinase gamma, when it's activated, it activates AKT and drives the proliferation of the macrophages and also this immune suppression phenotype. So you can inhibit that.
Other questions? This is a question for okay.
So I have a question about the last talk. So all of your expression patterns when you were presenting upfront the ARNT levels across subtypes and etc., they were pretty flat overall. So when you're doing this classification of first quartile versus last quartile, what is the separation that you have between the two quartiles that we're even looking at in ARNT?
Yeah, no, so we don't really see differences across, more closer, for example, like third quartile versus second quartile, so that's why we try to present the largest difference at first and the last. We don't really see such association exist.
And then when you're calling something as an alteration, does this mean that the transcript abundance was altered, or whether there was a mutation in the gene? What exactly are we defining as an alteration?
Yeah, no, so we don't really have those detailed information at this point, but those genomic alterations were detected either with the target panel by Caris or with the whole exome sequencing.
So I have a question for you also. So ARNT was first described perhaps because of its expression in endothelial cells. So do you know which cell types are expressing ARNT in your tumors, and could the expression level in tumor cells versus endothelial cells be deconvoluted and make a difference in your analysis?
Yeah, no, that's a great question. I don't really have a good be able to separate those out for you based on the list data we have here.
Well, I think the single cell will help. Other questions from the audience? For Yufei, strategy to try to deal with the immunosuppressive microenvironment? Have you considered encoding cytokines that can activate helper cells or change the immune suppressive myeloid cells?
Yes, so for us, our strategy is to armor the CAR T cells with ICI, and in the field there are, I think, USC South California, and they are doing the IL-12 different cytokines comparison compared to interleukin-15. I think there are so many different ways to conquer the immunosuppressive tumor microenvironment currently, even like shRNA armored CAR T cells to change the TGF-beta pathway in the CAR T cells. So there are different multiple paths.
There's a question back there.
Hello, question for Dr. Winayak. Our group at Dana-Farber has previously showed the importance of germline genetics in predicting immunotherapy toxicities. Specifically, IL-7 SNP is a biomarker that's predictive of immunotherapy toxicity. Simple question, did you look at that in your data set, and does it add to anything to whatever you presented today in terms of biomarkers?
Sure, thank you for your question. We didn't look at any germline or genomic data. It was purely looking at these baseline hematologic ratios. That might be quite interesting to see any correlates, and then certainly, looking at perhaps serial, markers, and then looking at how they link up. So, thank you.
So I have a question for you. What is the implication of the neutrophil to eosinophil ratio? So as a myeloid expert, that ratio that seems to be important is very curious to me. So.
Sure. So within the context of prediction of IRAs, I think we've shown, and actually other groups have shown as well, that low baseline NER seems to be predictive in that setting. I think the reason for that is eosinophils are known to modulate the immune system. They themselves can act as antigen-presenting cells. They can interact with dendritic cells, and I think perhaps that's probably a reason where we're seeing that it can be predictive. I do also know that low baseline NER can be prognostic as well. So, but I think in the context of of prediction of IRAs, it makes sense with the biology.
Thank you. And a final question for Dr. Hammers. Again, because of my expertise in the myeloid field, what I was wondering is whether the PTPN inhibitor can have an effect on the myeloid compartment that could be beneficial.
That I don't know. Alicia Emmons is an audience who may have some data on that. But it's certainly clear that we affect two compartments here. And I think the hope with reinstating interferon signaling is that we will make these tumors more susceptible to actually even the T cell cytotoxicity, etc., HLA presentation. I would be surprised if there was no effect on the MSDCs in that compartment, but I don't know. I don't know if Alicia.
There's a lot of crosstalk within the tumor microenvironment. There's very extensive dissection of the mechanism in the armor. We haven't looked in patients.
All right, well, if there are no further questions, I want to thank all the speakers for terrific work, and keep it up. So thank you all.
Thanks, everyone. Thank you, Wesley. Thank you.